The first topic is self-assembly. So I'd like to teach you all you need to know about self-assembly with this computer-generated model of a virus particle. Maybe you can see it better here. I've got it in a bottle. I apologize to those of you that have seen this before, but so basically, how does a virus put itself together? Well, this model is based on the PDB coordinates of the polio virus, but it's augmented, the subassembly units are augmented with magnets that represent the complementary electrostatic interactions along the edges of the viral subassembly. So this is not a single object. This is printed out of 12 identical objects with identical placements of those magnets. So I'm going to shake this, and what you'll see is that as I shake it, it starts to assemble. Why does it assemble? Well, it doesn't know anything about transformational matrices or anything else. All it knows about is geometry and the placement of forces. And as I shake it, what you'll notice is that the larger pieces stay together longer than the smaller pieces. But they also do break apart, but at a slower rate. Why is that? Because they make more interactions, and so the subassemblies are stronger. It's a survival of the fittest. So what is the most stable structure? It's the one that has all of the interactions made. Now, one thing I didn't tell you about this demonstration is that because it's stochastic, I don't know how long it will last. So it's a search problem, but it's taken care of by the physical nature of the objects themselves. So the point I want to make is that we have to look beyond our eyes in terms of how we visualize and communicate information. And technology is changing quite rapidly in that domain. I'd like to show you, again, a couple of demonstrations that use the physical models. These are models that are actually computer output, but use them in the context of an interactive system. And the interactive system is something that's coming along quite quickly, which is the use of augmented reality. In other words, there are smartphone apps now where you can hold it up to the sky and see the constellations and where they are. So interacting with the real world has a lot of benefit. The technology that we use to, let's see if I can get this to work, basically is an open source software called the AR Toolkit. And the way it works is that it recognizes in the video, threshold in video, the square. And because it can track the corners of the square, it gives you the X and Y coordinates. And because you know the size of the square, it gives you the Z coordinate. And if you tilt it, it gives you the orientation. So it gives you all 6 different degrees of freedom. If you have 2 of them, now I'm manipulating the model in the computer. I can calculate what the distances are, okay? Now, with the magic of computer printing, I can also make objects like this assembling peptide, the basic structure of the basic subunits of the proteins that Ed was talking about. I don't know if you can see that. So this is a physical model. So now, for instance, I put these virtual side chains onto my physical model. And so now I can manipulate the physical model, depending on the tracking here, and I can measure distances that are constrained by the physical model in the computer model. So this is a hybrid interface. And you can use this to look at all kinds of different types of interactions. This is another example on a different scale, and this is a protein-protein interaction. And here I've got the physical shape of superoxide dismutase, which is an enzyme that has an electrostatic field around it. And the electrostatic field guides harmful reactive species, negatively charged, down into, let's see if we can, now there always has to be a marker in view, right? So that's why I have several markers around this. So now you can see the electrostatic field lines that are pointing down to the active site of this enzyme, which takes care of these harmful charged free radicals. So the idea behind this is that in fact you can have all kinds of different physical objects that give you a certain kind of perceptual integrity that I won't go into in great detail, but the fact is that because you're using not only your visual senses, but you're using your proprioceptive senses and your sense of touch. One thing I was watching during all these visualization talks was the gesturing that the speakers are making. They are talking with their bodies, not just with their mouths. And so this idea of embodied learning, this idea that in fact integrating things using multimodal perceptual apparatuses is something that I think has some power in terms of conveying information. Now, I'll just show you one more demo, and this is just to show you that in fact, you can use this as a real interface for doing calculation. So here, for instance, I've got a small molecule, again, if I can get the recognition, and as I bring it closer in, you'll see in the histogram on the right, it's calculating the interaction energy between this small molecule and the protein as I manipulate it. Because I'm manipulating the physical object, the computer object that generated this physical object, essentially. So the idea of interfaces, I think, is an important one, and I thank the demo gods at this point for making this all possible. And now I'll talk about the work that is going on in the lab. And essentially, what I want to do is just make three very simple points about interaction. I got the directions about what the talk should be about, and it's supposed to be about interacting proteins, but I take it more to mean interacting with proteins. How is it that we can understand these complex structures? And those interactions take place on three different levels. There's the human interaction, and that is this physical, how is it that we get the most important distance, which is from the data into our brain and start to do something with it? How do we do that? And think perceptualization. Think about all of the things that make the world the world. I thank Tamra for the talk last night about the two-dimensional world. And she's absolutely right. Our eyes are made up of retinas that are basically like films. We got two of them, but that doesn't give us everything that's in the world. But the fact is that we do live and function in a three-dimensional world. That is the reality that we are dealing with. The data interaction, I think, is something that a conference like this should really focus on. I see a great divide between what I would term the string-based or informatic community and the three-dimensional community, and they are different types of information. But in fact, you need both to really understand what's going on. I like to use the analogy, if I gave you a connection diagram, a functional connection diagram of an automobile and asked you to build one, you might know how it works, but you certainly wouldn't be able to build one. You have to know physically what's going on. And so that's why I think this kind of data integration is so important. And finally, software integration. I mean, software is the life's blood of this community, but it's not community integrated, not at all. So those are the things that, you know, I'd like to kind of speed through if I could. But so the main point that I want to make is that we live in an age where there is a physical continuity between atoms to organisms. I mean, we don't need to invoke anything other than the physics and chemistry of what's going on in the biology. But that doesn't mean that it's not complex. Every level adds complexity. Physics to chemistry is made more complex by the combinatorics of the elements. And chemistry and biology is made much more complex by the fact that evolution embodies history into structure. So, you know, I won't go into the history of it, but in fact, crystallography has been a driving force for understanding the molecular world, and physical models have always been very important. But there was a great shift in the 70s between the physical model and the computer obviously won out. Just one side note, everybody knows how important the physical model was for the Watson-Crick double helix. Now, if you read the paper, there's not a single mention of that model in the paper. You have to read their memoirs to understand how important that model was. So what does that tell you? It tells you that the generation of ideas is not what's reported in the scientific literature. The most important thing in science is the generation of ideas and hypotheses, and then asking the right questions around those hypotheses. But that's not, and the physical model, I think, has a lot to do with it, or at least all of the cognitive aspects of our work. You know, so I don't have to go through this. I mean, biological structures are complex and so forth. The thing that I think is most interesting right now is this major shift in technology. Computers in everything, sensors in everything. I don't know how many of you have seen the new IBM Connect, but so that's a camera system that actually captures depth information, and it sells. You can buy it, you know, at the local Best Buy or whatever for about $150. This will change, and things like that, technologies like that will change how we interact with our computing environment. I mean, all of these gestures that we're making when we're talking can actually be put to good use. So that's the focus of the work that I just showed you, but I don't want to go into these details. I just have these in case the demo didn't work. But so these are the kinds of applications that we're interested in, and it goes from the simple translation of the linear information from the gene information into an actual working molecule, which is the protein folding, as the first step in that process. And protein-protein interactions are the major step of what is going on in biology. In other words, it's not the covalent bonding of the individual structures, it's the weak interactions that take place between all of the molecules. Drug design is an obvious area that once you know the structure, this is exceedingly valuable in terms of rational drug design, and we've been involved in that. David developed Autodock, which is the most popular docking code in the world about in 1990, so that's been evolving. But I want to talk about looking into the future in terms of how we might integrate this information into a larger picture of biology. And as you can see from all of the data, like the protein data bank and the EMBDB and all of the tomographic data and all of the fluorescence data, that needs to be integrated into a picture. There is no one instrument that is going to give you that picture. So David is the leader in kind of showing us what an integrated picture of the cell is from the watercolors that he has produced. But in fact, that's a first step in trying to actually generate a molecular view of the cell. This is David's view of a red blood cell in blood serum. Back in '97, I was inspired to try to just generate the same information as a 3D model. It took me a long time to do it, and that's with all the work that David did ahead of time. Graham Johnson came to the lab several years ago. He's a graduate student. He's about to graduate, and the work that I'm going to tell you about is his work. He came and wanted to do David's paintings in 3 dimensions. And so the first experiment that he did was basically, let's see if I can get this going. Okay. It was basically putting this inventory of molecules into a cell and then stirring it up, and then seeing if he could get the density of proteins in a cell from that kind of physical experiment. Now, he was using the tools that he, as a scientific illustrator who worked as an independent scientific illustrator and book illustrator for 10 years, used the high-end computer graphics tools that he was quite familiar with. And he was able to kind of do something, but he couldn't get the densities, actually, that David was representing in the cell. He also was interested in representing data across scales. So he went and worked with Brad Marsh in Brisbane for, I think, about a month or so, 6 weeks, and took Brad's cell data. Brad is actually has generated whole cell tomograms by mosaicing a bunch of individual tomograms together. I don't know why I'm having trouble starting these movies. Let me do it here. So this is Brad's data of a beta cell, which is a diabetes cell with granulocytes and so forth. And what Graham was able to do was to kind of systematize that so that he maintained the volumes of each of the elements so that you could see precisely how the components actually were distributed in different cells. So Brad actually did 2 activated beta cells and 2 inactivated beta cells, and they were interested to see what the differences were between the activated and the inactivated cells. And a typical pie chart did not show them some of the volumetric aspects that they were able to get from this type of representation. So Graham was well versed in this area when he came to my lab, and he was interested in developing tools that would actually fill up a cell with known complex geometries. And so basically, the idea here is to take, on the left, the organelle boundaries, things like the mitochondria and various cellular organelles, and take that data from the tomograms, and then take the data from the PDB of the known structures, kind of discretize the space, and then pack these things using an algorithm that he developed originally in Cinema 4D. So what he was able to do, I don't know why this is, I'll go back to the old technology here. So what he was able to do was basically to fill up space with shapes, both 3D spaces and 2.5D spaces, like manifolds or membranes and so forth. And he also had an algorithm to do stringy type structures in these spaces that you see in the chromosomal packing on the left. And and he could also specify gradients from points or planes and so forth. So all of these tools became available over the course of the time. And this is just one example of a movie. This is a synaptic cleft. This is data from Chris Arthur at Scripps tomographic data. And so Graham took that data and then basically cut out a volume. I have to keep. And using his fill algorithms, he filled it up. So these are the vesicles. These are the synaptic vesicles. The green particles are the glutamines or glutamates, and the spikes are the membrane phospholipids. And then, fill it up with the molecules. So these are just the proton pumps that are in the synaptic vesicle. So that's just a single protein in the structure. Now he adds all of the proteins that are just on the vesicle surface. So now what you're seeing are the vesicles and the proteins that are on the individual membrane surfaces. That alone is filling up most of the space, and that's not showing the cytosolic proteins that are in that volume. So this is the picture of that little piece of volume of the cell filled up with, one of those guys just flew by, I don't know where it came from, filled up with proteins whose structures we essentially now know. So the last thing that I want to mention is this idea. So that's the integration of data, and I think that it's very, very important. And this example of putting the informatic data into the physical environment, I think, is going to be very powerful once it's developed. The collaboration and the interaction between groups of people is what I want to talk about now, and that is the fact that, on the one hand, computer graphics has advanced to amazing levels, right? I mean, the movies that we see have everything in it. They have simulated behaviors. They have real, physics engines. They can do enormous kinds of things, but they don't know anything about molecules. So on the one hand, we've got, Hollywood that can do anything that they want with physical objects. And on the other hand, we have all of these powerful molecular modeling tools that can do anything that we want, we think, with molecules. But the problem is that these two communities are totally separated. The people that, especially the medical illustrators, so this is what the motivation of Graham, is that in fact, as a medical illustrator, he had a hell of a time trying to get that data into his Cinema 4D project, his environment, the one that he could work in to do the kind of animations that I'm showing here, and the kind of animations that you see in the poster sessions from Janet and Monica and the ones that you'll see tonight. So these kinds of animations this is what you do with Maya, this is what you do with Blender, this is what you do with Cinema 4D. You don't do it with PyMol, you don't do it with Raspberry, you don't do it with ARC software code. So the idea was, and the types of images that you see on covers these days. I mean, in the old days, I could get a cover just by, taking a picture off of my Evans and Sutherland line drawing display. But these days, I mean, you need to say something in your cover. I mean, these are editorial images. So this is an image that Graham did for a paper on the structure of a drug transporter. I mean, there it is. What can I say? And what I'm going to talk about briefly is work that just got published. I think I haven't seen the actual hard copy of it, but this is the cover of it on this embedded PMV, which is trying to bring these two kind of software together. And the idea behind this cover, which I think Graham was a stroke of genius, was that he took the molecules every paper that was in this issue is represented on this cover by the molecule that they're talking about. And he was able to put it together very quickly because of the ability to manipulate both the molecular and the kind of high-end graphical representations that he had available to him. And the way that he did that was essentially to marry the molecular modeling software that we've developed in our lab, and it's based on Python as the platform language. It's been developed by Michel Sanner and co-workers over the last 10 to 15 years, highly capable of understanding a lot of complexities of the molecular systems and the data structures that go along with it, and marrying it to the power and the capability of high-end graphics systems as they exist today and as they will continue to evolve. And that's the most important point. And so I won't go into the architecture of it, but essentially the idea is that since the Python molecular viewer was very much component-based, we could separate the viewing system from the molecular modeling system and take that and embed it. Since Blender and Maya and Cinema 4D all have Python interpreters, we could just embed PMV into that code and then develop that so-called API little stub that connects them together. And doing that, we have the capabilities of a full molecular modeling system in the environment of a high-end computer graphics and animation system that people use to make Hollywood movies and so forth. So you have all of the capabilities now inside. You don't have to take a PDB file, create a VRML file, import it into let's say, Maya. As a geometry, you can't identify a single atom in it because it's just a geometry at that point. So all of these things need to be meshed together. And I think that's one of the things that's going to make the future brighter if people develop software in this way. So, let me go back. So there are lots of different things that you can do, and the point is it's not just to make Hollywood movies. There's a lot of capability in these systems that could be used not only just for rendering, but for simulation and so forth. And in fact, because the molecular modeling system understands molecules, it can import molecular dynamics codes and other types of molecular codes into it. So you have a marrying of the two worlds. And so here I'm just showing you, so for instance, that this is this augmented reality system working in Cinema 4D, for instance. And this is a molecular dynamic system working with the augmented reality system in Cinema 4D. And these renderings are different. This is Pi Rosetta and a few other types of molecular dynamic systems, again, in the context of Cinema 4D or Blender and so forth. So, and here I'm just showing you kind of the thing that I showed you with the tangible models. Here, this is what you typically would do. Now, using the collision detection, you actually can't interpenetrate anymore. So here you're using and now you're using collision detection on each of the atoms, and it's fast enough so that you can do it in real time. So here you're using the power of the high-end graphic system to do more in terms of the molecular modeling. So the two things kind of are symbiotic to one another. And again, this autofill program, which Graham originally implemented in Cinema 4D, is now a plug-in to PMV, so it understands all of the molecular world. And in fact, this model was generated in the embedded PMV environment within Cinema 4D. So it was much faster than previous. So, and so what do you do with these complicated models? Well, you start to use them in simulations. This is just I mean, you'll see much better kind of dynamics later on, but the idea is that generating the environments themselves is important. And I won't show you this since we're running late, but I do want to acknowledge certainly the people in the lab, Graham Johnson and Ludovic Autain in particular, who have done tons of work on both the ideas behind melding these environments and actually implementing it and implementing the autofill algorithms and so forth. And David Goodsell and Michelle Sanner, who are stalwarts of the lab that actually, you know, do the work. And all of our collaborators. Now, thank you for your attention.